Hello everybody. Welcome to the Antibody Society webcast series on antibody validation. This is the second episode of the series where we look at how antibody validation affects the reproducibility crisis in biological sciences. The Antibody Society is an international non-profit association. We are a forum for facilitating discussion, disseminating information, providing guidance, and assisting in training and advancement of students and postdoctoral fellows. So if you care about antibodies, please join the Society. The speakers in this webcast series are experts from academia and industry, and they're expressing their independent assessments. They're not representatives of the Society. Viewers may write in questions at any time for the speakers to answer, and the speakers will remain online for 15 minutes at the end of the cast to answer any questions you may have. You can text in the questions at any time during the broadcast. In our first webcast, Andreas Pluktoon described the types of antibodies generally commercially available as research tools. We heard that only recombinant reagents may ensure future-proof reagent identification and reproducibility between experiments, but commercial antibodies are mainly not recombinant. This affects our efforts to reproduce experiments performed in other laboratories. I'm very happy to welcome Professor Glenn Begley, CEO of Biocurate in Melbourne, Australia, and Professor Cecilia Williams of KTH Stockholm, Sweden. Their teams have wasted much effort and money because of the non-reproducibility of commercial antibody reagents. Greetings, Glenn. Greetings, Cecilia. Thank you very much for joining us today. Glenn, some years ago, you and your colleague Lee Ellis wrote a startling report about your experiences as Vice President and Global Head of Oncology and Hematology at Amgen. Glenn, could you remind us of the problems you ran into there and the role of antibodies in these problems and the consequences?
Thank you. It's a pleasure to be with you here today. So for a period of 10 years between 2002 and 2012, I was the head of the Hematology Oncology Research Group at Amgen. And during that time, we attempted to reproduce some of the seminal findings in top-tier publications reported in the top-tier journals. And to our horror, found that we were unable to reproduce the majority of those findings. In fact, in about 90% of cases, we were unable to reproduce the findings. It was actually worse than that because our standard operating procedure was to go back to the investigators themselves and ask them to reproduce their experiments with an Amgen scientist standing next to them. And we were unable to do so, and they were unable to do so. It wasn't that it was a trivial finding in figure 2 that was not able to be reproduced. It was actually the major finding, the message of the paper, that was unable to be reproduced. Sadly, Amgen's experience is not unique. Bayer Health had reported it earlier, and then a number of groups have come out subsequently and reported that this is their experience too. So when talking about this, I think it's important to place this into some perspective. From my point of view, these results do not challenge the validity or the legitimacy of the scientific method. I'm not talking about fraud. What I am talking about is scientific laziness, sloppiness, ignorance, exaggeration, or desperation. I believe the vast majority of investigators do want to do the right thing. It's just that we have incentives that drive these sorts of behaviors. And the fact that this debate is occurring in public, in our scientific community, confirms the strength of our scientific system. Again, the fundamental problem is that we get what we incentivize, and while we incentivize publications in these top-tier journals, we will drive behaviors that result in papers in the top-tier journals. In addition, it's important to recognize that the advances that have taken place in medical treatment have been truly outstanding. So we have every reason to remain optimistic that research will continue to deliver. The sorts of treatments that are available today for cancer or for rheumatoid arthritis or inflammatory bowel disease were unimaginable when I was in medical school. So we've made enormous headway. The issue really is one of opportunity cost and the distraction that this is causing. And in my view, this could be impacted, if not solved, immediately by the funding agencies. So the sorts of criteria that I think are important in thinking about whether or not papers can be reproduced are listed on this slide here. And typically the papers that are unable to reproduce fail these half a dozen tests. Typically studies should be performed blinded. Sadly, that's seldom the case. Seldom are all of the results shown. Some of the worst offenders are Western blots where we see a sliver of a gel and we don't even see the full gel shown. Often experiments are not repeated. You'll be surprised how often in papers in the top tier journals a single experiment is reported. Seldom are positive or negative controls included. And specifically with respect to antibodies, reagents are often not validated. So immunohistochemistry is often performed with polyclonal antipeptide antibodies that are not legitimate for that purpose. And often the analysis of the data is not appropriate. So one example of a problem we stumbled into with antibody specificity is illustrated on the next couple of slides. So we were looking at antibodies that were allegedly detecting the erythropoietin receptor. And we took some of the commercially available antibodies that are shown on the panel here and used them to try and assess whether or not they detected the erythropoietin receptor. The example shown on the right of the screen is an illustration of what typically is shown in many studies where a sliver of a gel is shown and the other bands that are not indicated are just as important in terms of indicating the lack of specificity of this antibody. But investigators often show the result on the right to try and pretend that their antibody is actually specific. The erythropoietin receptor is a protein of 59 kilodaltons, indicated by the blue arrow. And you can see that the gels on the left have multiple bands present. Some of them are of great intensity, and certainly the dominant band is not at 59 kilodaltons. When we actually took that protein of 59 kilodaltons diagram there and did mass spec analysis, we found that the protein was not actually the erythropoietin receptor at all. It was heat shock protein 90 that shared a number of amino acids in common with the erythropoietin receptor. And because the reagent was a polyclonal antipeptide antibody, the antibody was very effective at pulling down both the heat shock protein 90, the dominant band, and the erythropoietin receptor as a consequence of those shared amino acids. The antibodies not only detect multiple proteins, as illustrated here, they're even able to allegedly detect the erythropoietin receptor in mice that lack the gene for the erythropoietin receptor. Of course, this is not the erythropoietin receptor, it's spurious binding, but it's indicated in the panel on the extreme left, where you can see apparently nice binding of the antibody to the mouse embryo that lacks the gene for the erythropoietin receptor. And you can also see an illegitimate control in the middle panel, where if one uses the peptide against which the polyclonal antibody was raised, you can effectively compete out the binding. This might make it appear that this is specific. Clearly, that's not the case. What we're actually competing out is one of the many other bands shown on the previous slide by using this peptide control. So this is not an example of specificity. It's actually an example of the peptide competing out all of the proteins that it would otherwise, the antibody would otherwise be able to detect. We published that result in 2006, and this same antibody can still be purchased, and in fact was used in a recent paper in Cancer Cell in 2015 to allegedly stratify women with breast cancer based on their expression of the erythropoietin receptor. This clearly, clearly is not an appropriate analysis. The erythropoietin receptor here is actually not the erythropoietin receptor at all. It's the same antibody that can detect the erythropoietin receptor in mice that lack the gene for the erythropoietin receptor, but does illustrate the challenge we have in taking commercially available antibodies and using them for, in this case, allegedly stratifying women with breast cancer. So that's a brief example of one of the stories that we stumbled into in terms of lack of specificity for antibodies that caused tremendous problems throughout the literature.
Thank you very much indeed, Glenn. So it's a major issue is the non-reproducibility of preclinical oncology research that you looked at could be traced to the inability to precisely identify, obtain, or reproduce the experimental conditions for crucial antibody reagents that other people used. I find that very depressing. Would you care to hazard a guess on how much time and effort that single example you told us about cost of a badly validated antibody? How much time and effort that cost your teams to find that out?
It's hard to be sure, but it probably took 5 or 6 people 2 or 3 years. So the initial experiments were first simply trying to validate all of the commercial reagents that were available, and we had to acquire them and test them. And then, having demonstrated that they were not fit for purpose, then we had to try and identify what the protein actually was that the antibodies were detecting, and that's when we stumbled across heat shock protein 90. It probably was 4 or 5 people's effort for several years to actually come to the conclusion. And then it's distressing that the same antibody continues to be used by well-meaning investigators who don't realize that the conclusions that they're drawing are fundamentally misplaced.
Yeah, dreadful. I'm sure Glenn has described generalizations that some listeners will think don't apply to their work. So let's get down to some specifics. Professor Cecilia Williams' team from the KTH Sweden generated some curious data while studying the estrogen receptor beta. Cecilia, welcome to the webcast. Thank you for joining us today. As I understand it, the estrogen receptor beta has been used as a biomarker in the therapy of breast carcinoma. Could you tell us about your experiences with antibodies to that receptor?
Yes, thank you for inviting me. That is correct. I would like to tell you a little bit about the background for the interest of estrogen receptor beta in breast cancer therapeutics and go on from there. So there is a blockbuster drug called tamoxifen, which has significantly improved survival for breast cancer patients, and it acts by binding and inactivating the estrogen receptor alpha. The problem is that about half of patients develop resistance. So when it was discovered in 1996 that we not only have one estrogen receptor, but also a second one, this one called estrogen receptor beta, there was a huge interest to see whether it was a possibility to improve endocrine therapy for breast cancer by either activating or inactivating this receptor. And there's been an intense research area now for 20 years with a lot of findings. And most in vitro studies have suggested that estrogen receptor alpha should be inactivated, but estrogen receptor beta should be activated. And because they have differences in their ligand binding domain, although both of them bind estrogen, one can also synthesize small molecules that inactivate or activate respective receptor. So it's an excellent therapeutic target for using in therapy. And based on this idea that estrogen receptor beta should be activated in breast cancer, several clinical trials have been initiated, and several other trials have been based on using estrogen receptor beta as a biomarker for good prognosis if it's upregulated due to treatment. However, there are some major problems in this research area. We have tried to study estrogen receptor beta in breast cancer for many years, but we could not find a single cell line that expressed this receptor from breast cancer. Then when the Cancer Genome Atlas came out with RNA-seq data for over 1,000 breast cancer tumors, we could clearly see that not breast cancer tumors, there was no real mRNA there either for estrogen receptor beta, while estrogen receptor alpha were there at high level as it was expected to be. And this begs the question whether we can have protein without mRNA, or whether there is some problems with the data that suggest that there are levels of estrogen receptor beta in a lot of breast cancers. When we compare the RNA and protein data, estrogen receptor alpha correlate very well with mRNA and protein, but estrogen receptor beta, we see no such correlation. This led us to look into the antibodies that are the basis for believing that estrogen receptor beta is expressed in breast cancer. We collected those that have been used mostly and are the most trusted antibodies in the field, 14C8 and PPG510, together with all other antibodies that we could get our hand on. And then we designed a validation scheme where we used very well corroborated positive and negative controls where we knew for sure that we had or did not have estrogen receptor beta expressed. And we put these controls to test with all the antibodies in using both immunohistochemistry, Western blot, immunoprecipitation followed by mass spectrometry to find which protein each antibody bound to. And then we compare their performance over all different human tissues using immunohistochemistry and correlated the staining to RNA as measured by RNA-seq. And this was done in collaboration with the Human Protein Atlas in Sweden. The validation scheme is detailed in more detail in a chapter published this year. And we already at the first step, we found that 12 out of 13 estrogen receptor beta antibodies did not perform as indicated in our positive and negative controls. In fact, only one could differentiate the positive and negative controls, and this was an antibody called PPZ0506, which had not previously been used in the literature on clinical tissues. Whereas the two most trusted antibody could not clearly differentiate, and the PPG510 also stained strongly in the negative controls. We also used a control for ER alpha through all methods to make sure that our method was working well. Next, when we used Western blotting, we could see that the PPZ0506 performed well in Western blotting, differentiating the positive from the negative control and also staining recombinant estrogen receptor beta, all of the right size, whereas the other two antibodies did not perform well. Using immunoprecipitation followed by mass spectrometry, we could see that the validated antibody bound to estrogen receptor beta, which we could detect by mass spec, but the other two antibodies, we could not find that they bound strongly to estrogen receptor beta, but they bound very well and reproducibly to some other nuclear proteins, which of course would give the wrong results when one uses this in immunohistochemistry, where we cannot see what size these other proteins has. And these are the antibodies that have been used to define the expression of estrogen receptor beta in breast cancer, whereas the validated antibody has not been used in clinical cohort. So this really puts into question whether estrogen receptor beta is really expressed in breast cancer. When we use these three antibodies over all normal tissues, 44 different human tissues, we could see that the validated antibody, here called PPZ, stained tissues where we could detect some level of estrogen receptor beta mRNA, although it's not highly expressed anywhere. Whereas the two other antibodies also stained all the tissues where we didn't have any estrogen receptor beta mRNA, and where our validated antibody did not give staining. And these tissues include breast, which we could not detect mRNA with RNA-seq, no staining with the validated antibody, whereas the two antibodies previously used in the field did give staining. Here is an example where we can see to the left normal tissues which express some estrogen receptor beta in a few cells. And to the right, we can see normal breast and breast cancer, where our validated antibody do not give any nuclear staining, but the two antibodies which have given rise to the notion that estrogen receptor beta is expressed in breast and breast cancer give a nice nuclear staining, similarly to what has been presented in the literature before, but we argue that this is not estrogen receptor beta. It's one of these other proteins that we have shown that this antibody can bind and stain. So the impact on the field of our studies is that we propose that a lot of the literature has been based on the wrong data, and that estrogen receptor beta is not expressed in all the tissues we believe that it is. And this can change our understanding of estrogen biology. Here is further examples how the validated antibody can stain where we have high levels of estrogen receptor beta mRNA, and it does not stain where we don't have mRNA, whereas the previously used antibodies stain in both cases. We postulate that the reason why clinical trials so far has failed when they try to activate estrogen receptor beta is because it's not expressed in the tissues that we think it is expressed in. This goes, for example, for breast cancer, where trying to activate estrogen receptor beta with estradiol, estrogen, has not given the result that it was expected to give. And the same thing for prostate. Using an estrogen receptor beta agonist has not generated the expected data, and we did not see staining of estrogen receptor beta in prostate either. The lessons we have learned is to really not trust everything in the literature or in the antibody that you can buy. Antibody validation is not simple. It takes a long time and a lot of effort, but the need for it cannot be overestimated. In this case, there is an extremely high cost of not having validated the antibody thoroughly. All in all, there has been 20 years of research in tissues where it seems like this receptor is not even present. There has been very costly effort by both small and large companies and multiple clinical trials, which cost a lot of money and also have a high cost in life quality. And with that, I want to thank everybody that has contributed to this antibody validation and important discussions relating to this topic. Thank you for listening.
Yes, thank you very much indeed, Cecilia. That's a tremendous story, and I think if we stop the validation series here, this would be enough information for people who have been paying attention to immediately go out and validate, need and want to validate any antibody that comes into their hands commercially. So thank you very much for this tremendous presentation. I think I should also note for beginners to the field what a phase 2 clinical trial costs. So just the financial burden, we're talking about somewhere between 15 to 50 million krona, dollars, euros, pick your unit, but in that sort of level. And of course, let's just briefly put to one side the human cost, but this is obviously money expended on something which is useless, which could have been expended on saving people's lives. One can look at it like that, just because antibodies were incorrectly described and the users trusted to some extent the validation with which they've been provided by the suppliers and didn't do the job of making sure the antibodies were fit for purpose, which Professor Williams' team so excellently did. And I think it's a tremendous example. Cecilia, yes, thank you for the tremendous work. And would you like to estimate, just guess, how many person hours your team wasted on discovering that the antibodies which were valid in the literature were had the completely different specificities from what was on the label?
Yes, we are talking not person hours, we're talking person years. We spent maybe 10 years studying this receptor in tissues where we now think it's not expressed. And then we spent several years doing the validation. A PhD student spent her major part of her thesis focused on this validation. So we are talking a lot of person hours.
Yes, what a huge amount of wasted wasted effort. And of course, you're an academic group, so you're funded out of public funds in the end, taxpayers' money, I guess.
Yes, which could come to better use.
Yes, absolutely. Thank you very much indeed, Cecilia. So today we've seen that both general and specific examples of why it's absolutely crucial to validate the reactivity of commercial antibodies you receive, and to do this in the precise experimental context that you're using. In other words, you have to make sure that the antibodies you receive are fit for purpose, fit for the purpose you intend to use them in. And remember this, what's written on the label is not necessarily correct. So, in short, you can lower the chances that you will contribute to the reproducibility crisis if you validate your antibodies. In this webcast, we heard from Professor Glenn Begley on how his team were unable to reproduce some 40% of the crucial experiments in oncology, often as a result of invalid antibodies. And Professor Williams showed how her team dredged through 13 commercial antibodies trying to find one specific for their target protein. In the next webcast, we'll be looking at how to find right antibodies for the job. After all, there are millions available. This is the Antibody webcast series on the validation of commercial tool antibodies. We're very grateful to Professor Williams and Professor Begley for joining us today, and we hope you'll join us next time. They will be remaining online for the next 15 minutes to answer any questions you have you would like to text in. Thank you for joining us, and we hope you'll be joining us next time for episode 3 in the series. The series would have been impossible without the generous financial support of our commercial sponsors, as shown on this slide. Until next time, from the Antibody Society.